Thank you for the introduction and for the opportunity to share our research with you today. I will be mostly presenting from our recently published manuscript led by Vanessa Porter, who is a very talented PhD student in our lab who is now working in the private sector. And I will be talking about human papillomavirus, or HPV, integration events in cervical cancer. So cervical cancer is driven by persistent infection with high-risk HPV subtypes, and in over 80% of cases, parts of the viral DNA become integrated into the human genome. Sites of HPV integration are associated with complex structural variation and dysregulation of the multiome, but this has been difficult to study using short-read technologies. To address this, we used long-read nanopore sequencing to characterize 72 cervical cancer samples from the HTMCP cohort. We previously characterized this cohort using short-read, so we do have matched whole transcriptome data available. And this is an overview of our cohort. You can see that we have the majority of the high-risk common HPV types. We also have patients with both HIV-positive and negative status. So using long reads, we were able to identify 6 different categories of HPV integration events, which we characterized based on their alignment patterns in the human genome. And the long reads also enabled us to resolve heterogeneous forms and copy numbers of the viral genome within individual integration events, with one example being shown on the lower left. So this is a single HPV integration event in the human genome, and you can see that there are various different configurations of the viral genome within this one integration event. As you all know, Nanopore is also able to detect base modifications, so we also looked at the methylation status of integration events. And I'd like to draw your attention to the bottom right-hand side here. So each row here represents one integration event, and the human genome is shown on the left, and the viral genome is shown on, right and then left. And we found that transcriptionally active integration events were interestingly characterized by demethylated regions downstream of the viral transcription start site, so downstream of the LCR, into the breakpoint between the virus and the human genomes, and the demethylation persisted on the human genome, leading to the production of HPV-human fusion transcripts, whereas transcriptionally inactive events showed inconsistent methylation patterns. Phasing was also mentioned previously as an advantage of nanopore sequencing, so we also looked at allele differences between the HPV-integrated allele versus the other allele, and we saw megabase scale differences in methylation on the HPV-integrated allele. This was also associated with allele-specific dysregulation of gene expression on the HPV-integrated allele, and on the far left side, we see one example in one patient. On the top, we see the HPV-containing allele, and you see that it's generally hypermethylated compared to the other allele, but there's this focal region of demethylation, and in particular, one differentially methylated region overlapping the second exon of this NRA43 gene. And in this particular sample, we also saw very high upregulation as well as allele-specific expression of this gene. So in summary, we used nanopore sequencing to resolve complex rearrangements of both the viral and human genomes at HPV integration events in cervical cancers. We saw a striking correlation between the local methylation and transcriptional status of HPV integration events, as well as distal allelic differences in both methylation and gene expression. I would like to thank everyone who was involved in this work, all of our lab, our collaborators in the Genome Sciences Center and also in Wisconsin, our funders, and of course the patients and their families, without whom this data would not be possible. Thank you for your attention. I'm happy to take questions.